अरू स्टुडेन्टहरु आउँदै गर्नुस् है त सुस्मिता सुषमा शिला ऋषभ रेजिना प्रीति लक्ष्मी कृतिमा ईशा देवका ल यतिजना हुनुहुँदो रहेछ लेट्स कन्टिन्यू है त अब आजको लेक्चरमा आजको मटेरियलमा है त सो बायोइन्फर्मेटिक्स लास्ट क्लास वी टक अबाउट के पढ्यो हामीले कोर्स कन्टेन्ट के छ भनेर वी डिस्कस्ड अब तपाईहरुलाई म एउटा चाहिँ आई वान्ट टु शेयर विथ यु अ कोट बाइ डेभिड नुथ right he was 1974 Turing award winner and what he has to say about biology either so I can be as confident about computer science as I can be about biology easily has 500 years of exciting problems to work on it's at the next level so David Nuth actually he's a computer scientist right a computer scientist le ke bhanirahnu bhacha cha bhandakheri that computer science might run out of exciting problems but biology it has like 500 years of exciting problems to work on it so that's something at the next level right so the question is can biological life be equated with computing now that's the major question that we have and the results so far would suggest that the answer is yes. हामीले के बुझ्नु पर्छ भन्दाखेरि के like हामीले last classes-हरुमा last semesters-हरुमा that we studied about biochemistry right we studied about chemistry and what was our conclusion that all living processes can be understood in terms of chemistry okay हैन त्यो logic त्यो conclusion निकाल्न सकिदैन biochemistry पढेर Hello? Devaka, you have something to say?
No, sir, but generally, धेरै कुराहरु चाहिँ सकिन्छ निकाल्नु.
Okay, right. Your analysis, your conclusion is that lots and lots of biological processes can be understood in terms of chemistry. है त अब अहिलेको मोडर्न जुन केमिस्ट्री छ त्यो मोडर्न केमिस्ट्रीको चाहिँ जुन मैले अहिले अजम्पसन गरिरहेको छु that is considered to be more or less a valid assumption. The valid assumption in मानिन्छ के like everything can be understood in terms of chemistry भन्ने किसिमको that assumption is considered to be very valid and in this course so my goal what will be my goal is that biological life can be equated with computing so we can understand lots and lots about life if we apply the rules of computing to the information biological information so हामीले अहिले पनि त्यही देखिरहेको छौं that lots and lots of things can be answered with the help of informatics or bioinformatics so there's a little kid right so he's thinking it in terms of ones and zeros ones and in terms of binaries and in the other sentence he's thinking in terms of ATGC the sequences the information of the DNA so bioinformatics essentially is the culmination of biology and informatics or the computer right so त्यो चाहिँ that is the basic crux of the matter right so हाम्रो के हुन्छ यहाँ intersection between biology and computer science so हामीले के देख्न सक्छौं there is this our genome मा के छ there is lots and lots of information right the information it can be about in terms of genetic information हुन सक्छ right and अब अहिले त हामी we also talk about the epigenetic information about like सबै informationहरुलाई चाहिँ we can encode in terms of computer र हामी के देख्न सक्छौं भन्दाखेरि here we have all these information ATGC information that can be encoded in some computational way so the modern era of biology needs to interpret a wealth of data tools that only computer science is able to provide and hence bioinformatics so हामीले के देखिरहेको छौं भन्दाखेरि right हामीले अहिले के देखिरहेको छौं भन्दाखेरि modern biology मा that there is wealth of information there is wealth of information of about sequence information from DNA or the RNA protein को पनि sequence information हुन सक्छ residues हरुको or different kinds of structural information है त interactions हरु right कुन प्रोटिनसँग इन्ट्र्याक्ट गरिरहेको छ, डिएनए प्रोटिनको इन्टरेक्सन, अल दिज आर इन्फर्मेसन्स. Basically they are informations, right? So the your information, like first ma ke huncha, this information they arrive in terms of data, and your data bata chai what we can do is data lai chai hami information ma convert garna sakchau, and with the help of bioinformatics tools we can make biological inference about these data, these informations. So हामी के बुझ्नु पर्ने भन्न छ भन्दाखेरि, so since biology generates lots and lots of data, that's why to understand this data we need tools to understand these data. So that's why we need bioinformatics. त्यो चाहिँ bioinformatics को एकदमै रोल छ. So कारण के रहेछ भन्दाखेरि, what is the main crux of the matter, that biology generates lots and lots of information, right? In various kinds of information. So we can have lots and lots of biological data, right? In the next class, we will study about computational approaches to biological problems. Then they will study about what are the different classes or the categories of the biological information that can be processed with the help of computer. So maybe these are the physical map, the genetic maps, chromosomes go, and this is a wealth of information. Now here we have wealth and wealth of information. So these all are information, right? As you can see. Okay, so these are all our informations, right? These are the chromosomes, right? In chromosomes, we have labeled the arm, right? But we have a different, fine, higher resolution. We have information about the location of genes. So all these information, right? It has to be converted into some form, and that has to be stored in computer. Computer ma store garnu parne huncha, right? So similarly we have अब हामी के छ यहाँनेरि डिफरेन्ट यहाँनेरि चाहिँ जिन्सहरु छ, right? अनि त्यसको चाहिँ हामी के देखिरहेको छौं, यसको चाहिँ यो सिक्वेन्सहरु छ, these are these different proteins हरु छ, right? यो different proteins हरु छ, these are different proteins. So your proteins को के छ, we have sequence information. The sequence information but we want to derive some kind of meaning, right? Isma, what is the meaning here, right? So even to understand this biological data, we need some kind of computational tool, because human mind, it cannot fathom these data. Human mind, it is very difficult to find meaning in this data. And we can see, like for example, here we can see. So you touch a computational tool. What it has done is that sequence alignment, multiple sequence alignment, and we can see different conserved regions, right? WPP, right? And here is another conserved regions R, right? So these are some of the conserved regions that we can identify, right? So this is one way to understand biological data with the help of bioinformatics. So this is our multiple sequence alignment, MSA, right? So similarly, we have lots and lots of structural data as well, all right? So we have a transmembrane protein, right? A very elaborate, very molecular weight एकदमै high भएको चाहिँ we have seen this data, this structure. यो स्ट्रक्चरलाई चाहिँ हामीले we have represented it, र यसलाई चाहिँ हामी कम्प्युटेसनली यसलाई चाहिँ हामीले represent गरिरहेको छौँ, यसको मोडलिङ गरिरहेको छौँ, right? This is the computer model of the this transmembrane protein. So यो पनि एउटा biological data हो. So यो biological data बाट चाहिँ we can have meaningful information. र कुन रिजन्सहरु कहाँनेरि कुन रिजन्सहरु छ, कन्जर्भ साइट्सहरु छ, राइट? कहाँनेरि एन्टिबडी कोलाई चाहिँ टार्गेट गर्न सकिन्छ, राइट? इफ इट्स अ भाइरल प्रोटिन, right? So all these things can be interpreted with the help of computational tools are used together. We can derive certain meanings, certain information from these data. So अर्को यहाँनिर के छ भन्दाखेरि we have these networks, है त? So यो एउटा नेटवर्क, पर्टिकुलर नेटवर्क हुन सक्छ, एउटा पर्टिकुलर प्रोटिनको हुन सक्छ, and how this particular protein is interacting with other proteins, right? So even all this is an interaction map, right? So some interactions are strong interactions, some interactions are weak interactions, right? So I mean, like the different way we can represent it in the computer we can represent it. So यहाँबाट हामी के देख्न सक्छौं, maybe we can find new information, right? Maybe this is a protein, right? यो चाहिँ एउटा चाहिँ certain एउटा चाहिँ cancer cause गर्ने gene हो, cancer cause गर्ने protein हो. And what happens if this is a protein, right? And what happens if there is mutation in this protein? Your protein ma mutation भयो भने के हुन सक्छ? Because since it is linked with other many protein, यसमा चाहिँ if there is a faulty, your enzyme faulty भयो भने or your protein faulty भयो भने चाहिँ it can impact a lot. So that way we can also generate therapeutics. है त कुनलाई टार्गेट गर्ने त, right? You target जहाँनिर चाहिँ maximum यसको चाहिँ impact होस्, right? So if this is a transcription factor, now you want to activate these transcription factors, right? So सबै players, right? यसको चाहिँ vertices छ, right? So maybe they are not all equally important. Some are more important than others, and we want to target those proteins. है त कुनै एउटा चाहिँ थेराप्युटिक ड्रग इन्टरभेन्सन गर्नुपर्ने छ भने we want to target those proteins. So कसरी थाहा पाउने भन्दाखेरि, so with the help of bioinformatics यसरी हामी चाहिँ we can make with the help of यसमा चाहिँ ग्राफ थ्योरी को, right, एकदमै ठुलो इम्पोर्टेन्सहरु छ. So even that graph theory can be applied to computational. So we can study biological interactions with the help of graph theory. यो चाहिँ mostly यो graph theory मा आउँछ है त. So graph theory हरु पढ्दाखेरि चाहिँ that will be more relevant. है त. अब यो कोर्समा चाहिँ हामी धेरै चाहिँ कम्प्युटेशनल को कुराहरु पनि पढ्छौं है त, because graph theory is one such domain of computer science that we will delve a little bit है त. हामी धेरै डिटेलमा delve गर्दैनौं, just a little bit. So similarly, what are the goals of bioinformatics? So the major goal of bioinformatics is that to manage data, right? So that because since a lot and lots of data are generated, and it's very easy nowadays to generate this data. Since we have lots and lots of tools, sequencing tools, right? We will also be studying about the next generation sequencing tools, right? The NGS tools, next generation sequencing tools, we will study, right? And then we will also study about some of the whole genome sequencing, well, WGS, whole genome sequencing tools. And these tools, these techniques, they can help us generate lots and lots of data, sequence data. So that's why हामीसँग के छ भन्दाखेरि त्यो डेटालाई चाहिँ इन्टरप्रेट गर्नुपर्ने हुन्छ, right? अब त्यो डेटालाई चाहिँ फर्स्ट we need to manage that data. डेटाबेस को लागि चाहिँ we have to design database. यो कसरी यो डेटालाई म्यानेज गर्ने भनेर त्यसपछि इजी एक्सेस पनि हुनु पर्यो त्यो डेटामा, right? And सबमिट गर्ने, रिट्राइभ गर्ने, त्यहाँ चाहिँ इन्फर्मेसन चाहिँ सर्च गर्ने त्यो टुल्सहरु हुनु पर्यो. And right, to analyze those biological data. Data itself has no meaning, है त. You have to understand that data itself has no meaning. It only has meaning when you analyze it, and you can analyze the data in different way, right? So what happens is that you can analyze data in a wrong way, and you can analyze the data in a biological way, biologically relevant way ma pani you can analyze. So त्यो पनि एकदमै important हुन्छ, right? So the bioinformatics tools, it will help you to analyze data and interpret the results from the biological perspective. So it's a very crucial thing. So in bioinformatics, we will analyze data from a biological perspective, right? हाम्रोमा चाहिँ जहिले पनि पर्सपेक्टिभ चाहिँ बायोलोजी हुन्छ. So यसमा चाहिँ हामी अलिकति कम्प्युटेशनल टुल्सहरु युज गर्छौं, कम्प्युटेशनल एल्गोरिदम्सहरु युज गर्छौं, and we will apply them in such a way that it makes a biological sense. So I mean, we can see that this is a map of an internet, है त. मैले अघि भनेको थिए. So map of the internet, and this is the map of human protein interactions, right? We have many proteins in our body, in our cells, right? And we are trying to simulate, we are trying to model the interaction that occurs between proteins and proteins, right? Proteins can be different protein, है त. Nucleus भित्रको प्रोटिनहरु पनि हुन सक्छ, ट्रान्सक्रिप्सन फ्याक्टर्सहरु, right? About proteins, it can be about cytoplasm, many different proteins, right? We studied about glycolysis, we studied about we studied about Krebs cycle, we studied about different biosynthesis, family podio. All those proteins, they interact with one another, right? And kuni proteins they interact with many other protein, right? So हामी यस्तै देख्छौँ, हामी इन्टरनेटमा पनि के देख्छौँ भन्दाखेरि there are certain locations, they are more bright, right? अब यसलाई चाहिँ हामीले के गरेको भन्दाखेरि अब यहाँ हामीले के गरिरहेको छौँ भन्दाखेरि we are trying to visualize this data. यो डेटा भिजुअलाइजेसन पनि इट्स अ भेरी इम्पोर्टेन्ट टुल, राइट? किन भन्दाखेरि त्यो डेटालाई चाहिँ त्यहाँनिर चाहिँ इजी इन्टरप्रिटेसनको लागि चाहिँ भिजुअलाइजेसन टुल्सहरू चाहिँ इज भेरी इम्पोर्टेन्ट, राइट? सो हामी के गर्न सक्छौँ भन्दाखेरि इफ अ सर्टेन नोड अर vertices has a lots and lots of activity, we can color code it in different color coding differently गर्न सक्छौँ, or we can make that size larger, right? We can make that brighter, right? Now there are different ways, right, to interpret that data, to visualize that data. So हामी के देख्न सक्छौँ भन्दाखेरि maybe, right, these two are operating at a diverse scale, है त? इन्टरनेट त एउटा चाहिँ नेक्स्ट लेभलमा अपरेट भइरहेको छ, राइट? सो यो त म्याक्रोस्कोपिक लेभलमा अपरेट भइरहेको छ इन्टरनेट, कनेक्टिभिटी अफ द इन्टरनेट, यो राउटर्सहरू, स्विचेसहरू, हब्सहरू, डिफ्रेन्ट कम्प्युटर्स एन्ड सर्भर्सहरू, राइट? सो यहाँनिर चाहिँ के छ, यो त एकदम न्यानो स्केलमा अपरेट भइरहेको छ. But what we can see is that there is lots and lots of similarities between these two. Like the key similarity of one possible reason is that they might are complex systems, right? They are both complex systems because they have lots and lots of interacting parts or the interacting components. So one goal of bioinformatics is to understand biological principles on genome-wide scale using the tools of bioinformatics, right? We want to understand, right? Actually, bioinformatics role के हुन्छ भन्दाखेरि to have a holistic approach of biology. Now this is in contrast to with the reductionist view that we had been continuing thus so far. Right. So again, so why we need computers? Because actually what happens is that since there are lots and lots of component, धेरै component हुने बित्तिकै के हुन्छ भन्दाखेरि our human mind is incapable of grasping, right? So के छ भन्दाखेरि यो कार्टुनमा what it has shown that so this guy since it's becoming too complex, he says that we need a computer because this system is getting out of hand. So that's why to understand this system, to understand the efficiencies of this system, we need to have a computer, right? So this can be a metabolic pathway, right? Different metabolic pathway in a cell. So, and what happens is that since we have gathered lots and lots of data, so since to understand this data, we need a human mind, right? So you take them as there is this dichotomy. So कुनै कुराको इन्फरेन्स निकाल्नको लागि चाहिँ you need to have a human mind or a human, है त? So हामीले यहाँ धेरै डेटाहरू जेनेरेट हुन थाल्यो, and just to find that meaningful conclusion, you need to have a human. So there is lots and lots of data out there in biology, but data without context, insight, and intuition of a human biological knowledge, computer and fancy program can only take us so far. That's why we need a computer, right, to understand more and more data. But what happens is that we have so much data, right, and even to have we need a context, we need an intuition of a human mind to understand this data from a biological perspective. The biological perspective, but we need a human mind. So this is a dichotomy, right? Bioinformatics ma, this is always this it's a balance between this intuition, insight, computer power. So there has to be a balance. Computational power it's not that we will get the solution. Right? So you need a balance between computational power as well as insight, wonderful insight, creative insight. So now let's define bioinformatics. According to National Institute of Health (NIH), bioinformatics is research, development, or application of computational tools and approaches for expanding the use of biological, medical, behavioral, or health data, including those to acquire, store, organize, analyze, or visualize such data. So overall, so bioinformatics concerns with biological data, and what we want to do with that data. So we want to acquire it, right? And we want to store it. So we'll be storing these in computers, in servers, and how we'll store these, right? So that it'll be easy to visualize it, to analyze it, to share it, right? To organize it. So there is also another related discipline called the computational biology. But computational biology is little different than bioinformatics in that. So computational biology is more mathematical in nature, and it will deal with the development and application of data analytical and theoretical methods. So mostly it deals with mathematical modeling, right? So mathematical modeling of life, right, or the biological system, and the computer simulation of living systems, techniques to study biology, behavior, and social systems. So actually, computational biology is a larger umbrella, and under that larger umbrella, bioinformatics is one of the subset of computational biology, because computational biology is more theoretical in its pursuit, because it deals with mathematical modeling and computational simulation uh, of the biological, behavioral, and social system. So yeah, different. It's already similar किसिमको छ है त bioinformatics र computational biology. So computational biology is more like भनिहाल्यो theoretical छ, बढी theoretical हुन्छ. So let's gather more definitions of bioinformatics. Right? So we have this National Human Genome Research Institute, NHGRI. So how has it defined bioinformatics? That is a branch of biology that is concerned with the acquisition, storage, display, and analysis of information found in nucleic acid and the protein sequence data. Right, यो दुईटा पनि एउटा चाहिँ डिफिनेसन्सहरू छ, right? These are the similar kind of definitions, है त. So Russ Altman and Dugan in 2003 has also offered two definitions of bioinformatics, right? The first involves the information flow following the central dogma of the biology, right? So हाम्रो के छ भन्दाखेरि there is this यो सेन्ट्रल डगमा छ, सेन्ट्रल डगमाको जुन इन्फर्मेसन फ्लो छ त्यो इन्फर्मेसन फ्लोलाई क्याप्चर गर्न खोज्ने, है त. So there is this information flow, right? DNA, RNA, protein, cellular phenotype, right? So the larger scale ma, while we are studying DNA को हामी चाहिँ study terms of genomics, RNA को transcriptome, protein को proteome, right? अनि cellular phenotype को cellular phenotype भनेर हामी चाहिँ will study. So यो पनि information छ, right? Genome मा, DNA मा information छ, RNA मा information छ, protein मा information छ. All that information we want to store it, display it, acquire it, right? Analyze it, right? So you first चाहिँ यहाँनेरि छ, है त. So first is the information, capturing the information flow following the central dogma of the molecular biology. And the second definition involves the flow of information that is transferred based on the scientific method. So what it involves? So यो चाहिँ के छ भन्दाखेरि this is a scientific एउटा चाहिँ methodology पनि हो, है त. So what you have to understand is that अर्को कुरा के पनि त you have to view is that bioinformatics भनेको एउटा मात्र कुरा होइन है. Bioinformatics भनेको एउटा चाहिँ साइन्स पनि हो, एउटा चाहिँ मेथड पनि हो, राइट. अनि अर्को भनेको चाहिँ यो चाहिँ एउटा टुल पनि हो. So this is a very different thing, है. The tool भन्नु र मेथड भन्नु, right? So it's a tool पनि हो, it's a tool, and as well as it's a method, way of thinking पनि हो, है त. So you have to be quite you have to be quite clear about this, right? It's both tool as well as method. So in the first the tool, right? And in second is a method. So it involves designing, validating, and sharing software, storing and sharing data, performing reproducible research workflows, and interpreting experiments. So these are our methods, right? How to carry out a successful bioinformatics research and यो के पनि हो भन्दाखेरि सो यो इन्फर्मेसन फ्लोलाई कसरी क्याप्चर गर्ने पनि एउटा चाहिँ साइन्स पनि हो हाउ टु क्याप्चर द इन्फर्मेसन द्याट इज इन द सेन्ट्रल डगमा So what we have is the first perspective of the field of bioinformatics, right? First perspective के हो भन्दाखेरि that we will be localized inside the cell. Cell मा के भइरहेको छ भन्ने कुरा मात्र we are concerned, right? And we have different kind of cell. Obviously, we have different kind of tissue systems. एउटा चाहिँ अब maybe the kind of information that is flowing in a liver cell will be certainly different from a muscle cell. So त्यो त फरक छ. But what happens is that all they follow the central dogma. So यो मा चाहिँ भएको इन्फर्मेसनहरु चाहिँ कसरी क्याप्चर गर्ने यसको मिनिङफुल इन्टरप्रिटेसन कसरी गर्ने भन्ने कुरा पनि that will be relevant. So bioinformatics has emerged as a discipline as biology has transformed by the emergence of molecular sequence data. So biology नै केमा ट्रान्सफर्म भइरहेछ भन्दाखेरि bioinformatics मा because there is lots and lots of data that is being generated. यो molecular data generate भइरहेको छ त्यो molecular data को लागि चाहिँ what we need certain tools, certain methods to interpret it. So this is the first perspective, right? And what is the second perspective is the organism perspective, right? यो little broad perspective छ, right? So organism मा के हुन सक्छ भन्दाखेरि there are three dimensions, है त एउटा time dimension छ with the time of development, right? एउटा चाहिँ region of the body कुन body को region हो र physiological or the pathological state, right? हामीलाई चाहिँ हाम्रो organism लाई it can be understood in terms of three dimensions. एउटा चाहिँ अब maybe it's a toddler or an infant or a fetus or an aging human being, right? So which part of the body are you talking about? Are you talking about the head or the eyes or the muscles or the limbs, right? And which kind of state is he in? So is he in a very normal physiological state or a pathological state, right? So even this can be even there is lots and lots of biological data, right? And we want to capture this data and we want to analyze this data with the help of bioinformatics. So it's a little bit of a broadening view, right? From the level of cell to the organism, and we consider the individual genome, collection of the genes, and including the genes that are expressed as RNA transcripts and the protein products. So here we have a little bit of broad view. And for an organism level, so bioinformatics tools can therefore be applied to describe changes to the developmental time, changes across the body regions, and changes in a variety of physiological or the pathological state. So even here we can apply the tools of bioinformatics. So what is the third perspective? So the third perspective is the perspective of that हामी चाहिँ like tree of life को perspective मा पनि हेर्न सक्छौं, right? First we saw the perspective of an individual cell, and second we saw the perspective of an individual organism, and third this is a very global view is that the tree of life, right? Of archaea, bacteria or eukaryotes भनेर पनि we can view life, right? And some are visible life and some are already extinct life, right? So the scope of bioinformatics includes all of the life on the earth, including three major branches of the bacteria, archaea, and the eukaryotes. For all species, the collection and analysis of molecular sequence data allows us to describe the complete collection of the DNA that comprises each organism, the genome. We can further learn the variations that occur between species and among members of the species, and we can deduce the evolutionary history of the earth, of the history of the life on earth. So हाम्रो के छ different pursuits हरु छ. We have three different pursuits. So this is the final pursuits. यसमा चाहिँ हामी के गर्न खोज्छौं भन्दाखेरि so overall हामी चाहिँ के छ त what are the variations between species and what kind of story can we develop it? The कस्तो किसिमको evolutionary story हामी चाहिँ develop गर्न सक्छौं. Like which organisms they came first and how they evolved into other organisms, र molecular level ma, right? Genetic level ma, what kind of changes, what kind of sequence changes brought these kind of variations, all that. Like your third perspective ma चाहिँ हामी tree of life को perspective एकदमै global perspective हामी चाहिँ लिन्छौं. These are the three perspectives of bioinformatics. So it's the same thing है त अब एउटै कुरालाई चाहिँ हामी we are trying to be redundant just so that you can better understand है त. So in a practical sense, so bioinformatics के रहेछ, it's about collecting, manipulating, analyzing and transmitting huge quantities of biological data, right? So यो नै कुरा हो. So if you have lots and lots of data, how so it's about collecting those lots and lots of data, manipulating, analyzing and transmitting those biological data. So यसमा के पनि हुन्छ भन्दाखेरि it's the application of computer technology to analyze and to manage large scale complex biological data. है त एकदमै use data लाई चाहिँ how to get certain insight from that data. So it's an interdisciplinary research area at the interface between the computer science and the biological science. So हामी के छ भन्दाखेरि we use different tools, algorithms that we can use from the computer science. है त computer science को एकदमै धेरै टुल्सहरु हामी युज गर्छौं. So bioinformatics is an interdisciplinary field that develops technologies that uses computers for storage, retrieval भनेको to download the information, right? Manipulation to data manipulation गर्ने, right? किन भन्दाखेरि why we need to manipulate the data? Because biological data has lots and lots of noise. है त manipulation किन गर्नुपर्छ? Because it has noise. यो noise कहाँबाट आएको हुन्छ भन्दाखेरि obviously so हामी junk DNA भन्छौं नि, maybe that junk DNA is a noise, some kind of noise. सबै information मा noise हुन्छ है त. All the information has noise. र त्यो सिस्टममा चाहिँ जहिले पनि त्यहाँनेरि biological system मा पनि त्यो noise लाई distinguish गर्न सक्ने ability हुन्छ, right? And distribution of the information related to biological macromolecules such as DNA, RNA and protein. So it combines computer science, statistics, mathematics and engineering to analyze and interpret biological data. So as technology, bioinformatics is a powerful technology to manage, query and to analyze big data in life science, right? अब हामी चाहिँ यो big data भन्छौं नि, right? So यो big data, so we'll talk about big data, data mining, है त, that are the essential part of bioinformatics. So your technology, this is the technology. So as a technology, you as a methodology, bioinformatics is a top down, right? Top down technology. It's a holistic, data driven, genome wide and systems approach that generates a new hypothesis, finds new pattern and discovers new functional elements. So what you have to understand is that, so you have to understand that technology पनि हो, right? It's technology as well as a methodology पनि हो. So methodology मा के हो, यो चाहिँ holistic approach हुन्छ यसको, right? And data driven हुन्छ र we will generate new hypothesis, we will find new patterns and we will discover new functional elements. So in bioinformatics has been used for the in silico analysis of the biological queries using mathematical and statistical techniques. So एकदमै statistics को चाहिँ एकदमै important role हुन्छ है त, because to have that relevant है त confident information को लागि चाहिँ we always need that statistical tools हरुको चाहिँ जहिले पनि we need that mathematical and statistical techniques हरु पनि हुन्छ. So in silico भनेको के हो, on the computer है त. हामी के गर्छौं भन्दाखेरि एभ्री मोडलिङ केमा गरिरहेको हुन्छौं, कम्प्युटरमा गरिरहेको हुन्छौं. सो हामी के गर्छौं भन्दाखेरि बायोलोजिकल डेटालाई चाहिँ विल कन्भर्ट एन्ड विल सेभ विथ दैट डेटा इन द कम्प्युटर चिप्स है त. अब सबै डेटाहरु चाहिँ कम्प्युटरमै सेभ गर्ने हो, सिक्वेन्स इन्फर्मेशन्सहरु देखि लिएर स्ट्रक्चरल इन्फर्मेशन्सहरु सबै हामी त्यसलाई चाहिँ कम्प्युटरमा सेभ गर्ने हो र त्यसमा सेभ गरेपछि हामी त्यसलाई चाहिँ सिमुलेसन गर्ने हो. सिमुलेसन गरेपछि what kind of biological insights we can attain either. So in silico is an expression used to mean the performed on the computer or via computer simulation. So I'm computer simulation गर्न सक्छौं, right? So the primary goal is to increase the understanding of the biological process, right? So they पनि understanding गर्ने यही हो, and it focuses on developing and applying computationally intensive techniques to achieve this goal. And के हुन्छ यसमा, so we'll study about the pattern recognition, right? Pattern recognition एकदम important role हुन्छ. Data mining, the big data मा धेरै डेटाहरु छ भने we want to have that एकदम relevant information. To noise र signal मा चाहिँ we want to distinguish, right? यो चाहिँ अब नयाँ नयाँ algorithms हरु, machine learning algorithms हरु and visualization techniques हरु, right? So these are some of the computational techniques, है त, कुन टेक्निक्सहरु छ प्याटर्न रिकग्नाइजेसन एकदमै हो। त सिक्वेन्सहरुमा चाहिँ कस्तो कस्तो किसिमको चाहिँ प्याटर्न्सहरु छ, राइट? यो प्याटर्नले के कुन बायोलोजिकल मिनिङ क्यारी गरिरहेको छ, राइट? यो प्याटर्नमा चाहिँ कुनै ट्रान्सक्रिप्सन फ्याक्टर आएर बाइन्ड गर्नको लागि बनेको हो, राइट? सो एउटा प्याटर्न के हुन सक्छ भन्दाखेरि टाटा बक्स, राइट? सो टाटा बक्स के हुन्छ, एक्ज्याक्ट सेम हुन्छ सबै सिक्वेन्समा। टाटा बक्सको के हुन्छ, स्टक टाटा बक्सको के हुन्छ, सिक्वेन्स एक्ज्याक्टली सेम हुन्छ सबैमा। एकजनाले भन्नुहोस् न मलाई। एकजना एनीबडी के हो टाटा बक्स भनेको के हो? अनि हेलो, सुनिरहनु भएको छ तपाईहरु मैले भनेको?
सुनिरहेको छ सर।
टाटा बक्स भनेको के हो? व्हाट इज टाटा बक्स? भन्नुहोस् त के हो टाटा? अब प्रमोटर सिक्वेन्स हो, राइट? मैले यो टाटा बक्सको कुरा किन मैले भनिहालेँ, मैले अघिदेखि भनिरहेको छु, मैले अघिल्लो क्लासमा पनि भनेको थिएँ कि मोलिकुलर बायोलोजी को चाहिँ तपाईहरुलाई अन्डरस्ट्यान्डिङ हुनुपर्छ है त। इफ यु डोन्ट ह्याभ द अन्डरस्ट्यान्डिङ अफ मोलिकुलर बायोलोजी then it will be very difficult for you to understand bioinformatics, है त? किन भन्दाखेरि अब क्याल्कुलेटर नम्बर नै नचिनेको मान्छेलाई क्याल्कुलेटर दिएर केही रोल हुँदैन नि त। सो द्याट यसको काम चाहिँ के हुन्छ भन्दाखेरि जस्ट टु एड यु, है त? यो कम्प्युटेसनल बायोलोजी अर बायोइन्फर्मेटिक्सको रोल भनेको चाहिँ एड गर्नलाई मात्र हो। अब यहाँ यस्तो हुन्छ है त, टाटा बक्सहरु जुन छ, राइट? यो प्रमोटर सिक्वेन्सहरु, यो सिक्वेन्स अफ डीएनए छ, है त? प्रमोटर रिजनमा हुन्छ, यो एउटै हुँदैन, है त? यसको सर्टेन एउटा चाहिँ कन्सेन्सस सिक्वेन्स भन्छ क्या हामी, है त? एउटा पर्टिकुलर सिक्वेन्सहरु हुन्छ, एक्ज्याक्ट त्यही हुँदैन, कुनै कुनैमा फरक हुन्छ, कुनै कुनैमा ए हुन सक्छ, कुनै कुनैमा टी हुन सक्छ, अनि कुनै कुनैमा टी छ भने कुनै कुनैमा ए हुन सक्छ, राइट? सो त्यो त एउटा प्याटर्न जस्तो। अब मलाई चाहिँ के छ भन्दाखेरि एक्ज्याक्ट त्यही हुँदैन। मैले तपाईलाई नयाँ जिन दिएँ, right? And now I want to tell you identify the promoter region here, the Tata box here. अब एक्ज्याक्ट सेम त हुँदैन नि त। अब तपाईले के गर्नुपर्छ भन्दाखेरि त्यो सिस्टम यसरी डिजाइन गर्नुपर्छ, राइट? सो कसरी डिजाइन गर्नुपर्छ भन्दाखेरि प्याटर्न चाहिँ रिकग्नाइज गरियोस्। किन भन्दाखेरि टाटा बक्सको एउटा सर्टेन प्याटर्न छ, एक्ज्याक्ट त्यही प्याटर्न हुँदैन, देयर इज सर्टेन प्याटर्न। अब त्यो प्याटर्न चाहिँ तपाईले यु हेभ टु आइडेन्टिफाई। सो त्यसलाई चाहिँ हामी के भन्छौँ, वी कल इट प्याटर्न रिकग्निसन। डेटा माइनिङ भनेको पनि त्यस्तै हो। डेटा माइनिङ चाहिँ इट एप्लाइज इन द लार्जर डेटा, एकदमै धेरै डेटा छ भने चाहिँ वी एप्लाई द डेटा माइनिङ। अब मेसिन लर्निङ अल्गोरिदम्सहरु छ, यो एमएल अल्गोरिदम्सहरु, राइट? अब यसमा चाहिँ अब हाउ टु टिच द कम्प्युटर, है त, अर द अल्गोरिदम्स टु लर्न इटसेल्फ, है त? नयाँ नयाँ कुराहरु लर्न गर्न कसरी सिकाइन्छ, है त? बिकज त्यस्तो पनि वी क्यान जेनेरेट अल्गोरिदम्स द्याट क्यान लर्न इटसेल्फ, है त? सुपरभाइज लर्निङहरु गरेर चाहिँ हामी वी क्यान टिच अल्गोरिदम्स टु फाइन्ड थिङ्स, है त? हामी विल टक अबाउट अब यो मार्कअप मोडलहरुको बारेमा पढ्छौँ, है त? एन्ड द्याट मार्कअप मोडलहरुमा चाहिँ यो मेसिन लर्निङमा एकदमै युज हुन्छ, है त? यो मार्कअप मोडल्सहरु कसरी हाउ टु टिच, है त? कम्प्युटरलाई चाहिँ यो चाहिँ लर्न गर्न, हाउ टु टिच द अल्गोरिदम टु लर्न इटसेल्फ एन्ड भिजुअलाइजेसन, डेटा भिजुअलाइजेसन। जस्तै अघि हामीले यो सबै अगाडि गर्यौँ, दिस इज अल अबाउट द भिजुअलाइजेसन, राइट? यो भिजुअलाइजेसन हाम्रो, है त? दिस इज हाउ वी आर भिजुअलाइजिङ द बायोलोजिकल डेटा। सो यो भिजुअलाइजेसनको पनि एकदमै ठुलो इम्पोर्टेन्ट रोल हुन्छ। Analyzing biological data to produce meaningful information involves writing and running software programs that uses algorithms from graph theory, right? एकदमै इम्पोर्टेन्ट हो बायोलोजीमा अहिले एकदमै ग्राफ थ्योरी एकदमै इम्पोर्टेन्ट छ सिस्टम्स बायोलोजीमा बायोइन्फर्मेटिक्समा ग्राफ थ्योरी एकदमै रिलेभेन्ट छ राइट इन्टरेक्सन्सहरू पत्ता लगाउनको लागि एकदमै अब यो कुरा चाहिँ यु हेभ टु अब स्टडी है त्यसलाई यसरी बुझ्नुपर्छ ग्राफ थ्योरी वाज नेभर created to study living systems okay na graph theory the living systems को लागि बुझ्नको लागि त बनाइएको थिएन नेभर र हामी के देखिरहेको छ भन्दाखेरि ग्राफ थ्योरीको प्रिन्सिपल्सहरू दे आर एक्सट्रिमली युजफुल टु स्टडी बायोलोजिकल सिस्टम्स है त त्यो भएर वी हेभ टु स्टडी ग्राफ थ्योरी आर्टिफिशियल इन्टेलिजेन्स राइट सफ्ट कम्प्युटिङ डेटा माइनिङ राइट सिग्नल प्रोसेसिङ image processing and computer simulations all these with the bioinformatics ma all these एकदमै ठुलो यसको इम्पोर्टेन्स हुन्छ है त यो अल्गोरिदम्सहरुको so bioinformatics derive knowledge from computer analysis of the biological data these can consist of information stored in the genetic code right experimental result from the various sources right so अब यो 2D gel electrophoresis को डेटा हुन सक्छ right gene expression को चाहिँ डेटा हुन सक्छ है त डिफरेंट एक्सपेरिमेन्ट्स हरुको डेटा हुन सक्छ so patient statistics हरु हुन सक्छ right and scientific literature अब all these data हरुलाई चाहिँ के गर्नु पर्यो so storage retrieval analysis सबै है त यो बायोलोजिकल डेटा हरुको so different levels or the different classes of the biological data हुन सक्छ so the relationship between computer science and biology is the natural for the several reasons के कारणले it's natural because the phenomenal rate of biological data being produced provides challenges massive amount of data have to be stored analyzed and made accessible यो accessibility को पनि एकदम important हुन्छ so that मैले generate गरेको data I want it to be available to everybody है त because science only works when there is some kind of collaborative effort यो accessible बनाउनको लागि चाहिँ there has to be some computational softwares right the computational softwares are हुनुपर्छ so that large amount of biological data can be transferred from one place to another right so the nature of the data is often such that a statistical method and hence computation is necessary biologically meaningful data पनि generate गर्नको लागि चाहिँ you have to be sure है त डेटा मा नोइज कति छ डेटा मा सिग्नल कति छ भनेर चाहिँ you have to have that statistical method है त स्योर हुनको लागि we will study about these statistical methods है त पछि हामी यो sequence alignment हरु गर्दाखेरि we will see some of the statistical methods that needs to be applied so that we can be sure about our match so this applies in particular to the information on the building plans of proteins and of the temporal and the spatial organization of their expression in the cell encoded by the DNA so different किसिमको statistical tools हरुबाट चाहिँ we can validate our data valid गर्न पनि we can be it can be helpful and there is a strong analogy between DNA sequence and a computer program so there is lots and lots of similarity the अघि मैले भनिहाले computer program भनेको ones and zeros मा it works right अब यो DNA sequence पनि ATGC मा छ right ATGC so त्यो भएको कारण there is lots and lots of similarity between computer science and biology so त्यो भएको कारणले पनि यो चाहिँ यो जुन union छ of the biology and computer it's very interesting so let's study about bioinformatics पढ्दाखेरि let's study a little bit about genomics किनभने bioinformatics को एउटा पार्ट पनि genomics पनि हो so genomics is the study of the genome which is the complete set of the genetic material of the DNA present in the organism so genetics मा चाहिँ हामी के गरिरहेको हुन्छौं भन्दाखेरि एकदमै स्मलर कम्पोनेन्टहरु मात्र स्टडी गरिरहेको हुन्छौं जिन्सहरु जिनको इन्टरेक्सन्सहरु मात्र स्टडी गरिरहेको हुन्छौं but while we are studying the genome we are studying it in the यसको चाहिँ entirety मा चाहिँ स्टडी गरिरहेको हुन्छौं एकदमै होलिस्टिक अप्रोच लिइरहेको हुन्छौं right so it studies all the genes and the interrelationship in the organism so as to identify their combined influence on growth and development so the aims of the genomics are extremely higher. एकदम लफ्टी गोल्सहरू हुन्छ जेनोमिक्समा, तर के छ भन्दाखेरि यो लफ्टी गोल्स मात्र भएर हुँदैन, त्यो लफ्टी गोल्सलाई एटेन गर्नको लागि टुल्स एन्ड टेक्निक्स मेथोलोजिजहरू पनि हुनुपर्छ, राइट? र हाम्रो अहिले के देखिरहेको छ भन्दाखेरि सो यो लफ्टी गोल्सहरू हामी एचिभ गर्न सकिरहेछौँ किन भन्दाखेरि बिकज देयर इज दिस राइज इन कम्प्युटेसनल पावर। कम्प्युटेसनल पावर पनि हामीले के देखिरहेको छ भन्दाखेरि देयर इज ग्रोथ। आजभन्दा 30 वर्ष अगाडिको कम्प्युटरको जुन पावर छ प्रोसेसिङ पावर र अहिले जति प्रोसेसिङ पावर छ द्याट इज लाइक डे एन्ड नाइट डिफरेन्स। र यो एकदमै जुन लफ्टी गोल्सहरू पनि हामी अटेन गर्न सक्छौँ बिकज द्याट देयर इज लट्स एन्ड लट्स अफ कम्प्युटेसनल पावर। त्यो भएको कारणले जुन लट्स एन्ड लट्स अफ जुन डेटा पनि जेनेरेट भइरहेको छ इभन वी क्यान मेक सेन्स अफ द्याट डेटा। So the field of genomics attracted worldwide attention in the late 1990s with the race to map the human genome, right? So 1990s what we did is that so human genome project हामी start भएको थियो, right? It was a big project. Biology मा अहिलेसम्म भएको सबैभन्दा लार्जेस्ट प्रोजेक्ट त्यही थियो, एकदमै एक्सपेन्सिभ प्रोजेक्ट थियो, अराउन्ड $3 billion खर्च भएको थियो त्यतिखेरको पैसा, राइट? It was a big amount and एकदमै धेरै डेटाहरू जेनेरेट भएको थियो। त्यो डेटा जेनेरेसन भएको डेटालाई चाहिँ लाइक त्यो जेनोमिक लेभलको डेटा, जेनोमिक्स लेभलको डेटा जेनेरेट भएको थियो। त्यो डेटालाई चाहिँ मिनिङफुल्ली इन्टरप्रेट गर्नको लागि पनि जेनोमिक्सको चाहिँ डेभलपमेन्ट भएको थियो, राइट? बायोइन्फर्मेटिक्स को डेभलपमेन्ट भएको थियो। द ह्युमन जिनोम प्रोजेक्ट वाज कम्प्लिटेड इन अप्रिल 2003, मेड अवेलेबल फर द फर्स्ट टाइम द कम्प्लिट जेनेटिक ब्लुप्रिन्ट अफ द human being, right? So in 2003 the complete, more or less complete है त, more or less भनिरहेको छु, so complete चाहिँ एउटा चाहिँ sequence of information it was generated है त। अब त्यो जेनेरेटेड भएको डेटालाई के गर्नु पर्यो, it has to be stored है त, स्टोर गर्नु पर्यो, त्यसलाई चाहिँ अब इजी रिट्राइभल हुनु पर्यो, त्यहाँबाट चाहिँ मिनिङफुल इन्फर्मेसन्सहरू चाहिँ ग्यादर गर्न सक्नु पर्यो। सो द्याट इज वान अफ द च्यालेन्जेस अफ बायोइन्फर्मेटिक्स। So genomics पछि we have this proteomics, right? So it's the large scale study of the proteomes, right? All the proteins, all the transcription factors, right? के छ प्रोटिन्सहरू हाम्रो चाहिँ सेलमा। So it is the set of proteins produced by the organism, system or the biological context, right? So we may refer for example proteome of the species सबै के छ, होमो सेपियन्समा कुन प्रोटियोम्सहरू चाहिँ बनिरहेको छ and how they are interacting। सो एउटा अर्ग्यान लेभलमा पनि हुनसक्छ, लिभरले के प्रोटिन्सहरू एक्सप्रेस गरिरहेको छ, इभन यु क्यान वर्क एट अर्ग्यान लेभल अर द स्पिसिज लेभल, बट प्रोटियोम इज नट कन्स्ट्यान्ट, राइट? इट डिफर्स फ्रम सेल टु सेल एन्ड चेन्जेस ओभर टाइम। डेभलपमेन्टल स्टेज अनुसार चाहिँ प्रोटियोमको लेभल, प्रोटियोमको चाहिँ डिस्ट्रिब्युसन चाहिँ डिफ्रेन्ट। सो टु सम डिग्री द प्रोटियोम रिफ्लेक्ट्स द अन्डरलाइन ट्रान्सक्रिप्टोम, राइट? कस्तो कस्तो किसिमको चाहिँ अब मेसेन्जर एमआरएनएहरू चाहिँ जेनेरेट भइरहेको छ, टिआरएनएहरू कति छ, राइट? सो ट्रान्सक्रिप्टोममा चाहिँ एभ्रिथिङ चाहिँ अब रिलेटेड टु आरएनएको चाहिँ ट्रान्सक्रिप्टोममा आउँछ, राइट? सो द सम टोटल अफ द इन्टरेक्सन्स अफ द दिज RNA. So however, protein activity is also modulated by many factors in addition to the expression of the relevant gene. So in proteomics, we study about all the effects. So it was a large scale study of the proteomics. So even this requires lots and lots of computational power. एकदम धेरै अमाउन्ट अफ इन्फर्मेसन छ है त प्रोटियोमिक्समा, कसरी एउटा प्रोटिन अर्को प्रोटिनसँग इन्टरेक्ट गरिरहेको छ, राइट? अप रेगुलेट गरिरहेको छ कि डाउन रेगुलेट गरिरहेको छ कि, राइट? एन्ड हाउ टु क्याप्चर दिज इन्फर्मेसन इन अ कम्प्युटरमा कसरी क्याप्चर गर्ने यो इन्फर्मेसनलाई, राइट? सो अघि मैले भनिहालेँ, सो एउटा एप्रोच के हुनसक्छ, वी क्यान अन्डरस्ट्यान्ड, वी क्यान युज द हेल्प अफ द ग्राफ थ्योरी। So proteomics, we can study about proteomics, when and where the proteins are expressed, right? The rates of protein production, degradation, and the steady state abundance. How proteins are modified, post-translation modifications, phosphorylation, right? So the movement of protein between subcellular compartments, the involvement of protein in the metabolic pathway, and how these proteins they interact with one another, right? So all information, we can generate. So which protein interacts with a particular protein of interest, right? अब यो ट्युमर सप्रेसन जिनहरू छ P53 भन्ने जिन, यो P53 जिनमा चाहिँ कुन जिन चाहिँ कुन चाहिँ प्रोटिनले चाहिँ इन्टरेक्ट गर्छ, राइट? त्यो हुन सक्छ, or which proteins are localized to a subcellular compartment, right? अब माइटोकन्ड्रियामा कुन प्रोटिन्सहरू छ त्यो पनि हुन सक्छ। हाम्रो एउटा चाहिँ इन्फर्मेसन क्यान जेनेरेट दिस काइन्ड अफ बायोलोजिकल इन्फर्मेसन, or which proteins are involved in the biological process. For example, in the circadian rhythm. Circadian rhythm मा कुन प्रोटिनहरू इन्भल्भ गर्न सकिन्छ, कुन प्रोटिन्सहरू इन्भल्भ भइरहेको छ. We can have this kind of information from the proteomics. So what happens is that there is this big biological data. Also challenges and then there is this opportunities, right? Every level there is lots and lots of information, right? You can see in genomics there is so much information at this genome level, right? Nucleotide sequence information, right? And then we have this gene expression information, right? Not all the genes are expressed in all the tissue types. Some genes are expressed during the fetal conditions, some genes are expressed during other conditions, during different environmental stress, right? All that, it has lots and lots of information. And there is this protein sequence information, right? And there is the information on protein families, motif, and domains, right? And there is this protein interaction. Different proteins are interacting with one another, and there are pathway information, and then are all these system information, different layers and layers of information, hierarchies of information, how to analyze them, how to organize them, how to gather meaningful data from them, meaningful information from them. So there is challenges as well as opportunities, right? So in the omics era of the life sciences, the data is represented in many forms, so which represents the information at the various levels of the biological systems, right? Various level, DNA level, protein level, tissue level, right? So many pathways level, systems level. So it can be the genome level, transcriptome level, epigenome level, proteome level, metabolome level, image processing data, molecular pathways data, different population of people, clinical and the medical records. All these are the different kinds of records. So the biological big data is big, and its scale has already been well beyond the petabyte or even the exabyte, right? Lots and lots of data. So this biological data will create huge amount of values if scientists can overcome many challenges. So what are the different challenges, right? Challenges are how to handle the complexity of the information, how to integrate the data from the heterogeneous sources, and what kind of principles or the standards to be adopted when facing the big data, right? So this is the major challenge either. So the tools and the techniques for analyzing big biological data enables us to translate massive amount of information into a better understanding of the basic biomedical mechanisms. So it all depends, right, how we understand these data. कसरी हामीले यो डेटा जेनेरेट गर्ने. यहाँसम्म सबैले बुझिरहनु भएको छ? Everybody is clear up to this point? सबैजनाले ल है त म तपाईँ ओके अब ओके म तपाईँलाई एउटा चाहिँ आई वान्ट टु सेयर विथ यु अ स्टोरी है त. यो स्टोरी के छ भन्दाखेरि तपाईँहरूले सुन्नु भएको छ, एउटा चाहिँ हात्ती आएछ एउटा गाउँमा, राइट? एलिफेन्ट, एन्ड देयर आर दिस ब्लाइन्ड पिपल एन्ड देन यो ब्लाइन्ड मान्छेहरूले चाहिँ के गर्छ, त्यो हात्तीलाई चाहिँ छाम्छन् है त, हात्तीलाई छाम्छन् है त. त्यो मात्र छ उनीहरूसँग, त्यो छाम्ने मात्र त्यो सेन्सरी मेकानिजम छ. र अब एउटाले चाहिँ अब चाहिँ पुच्छर समाते छ, एउटाले चाहिँ अब कान समाते छ, एउटाले चाहिँ खुट्टा समाते छ, एउटाले चाहिँ त्यो बिग एब्डोमेन रिजन समाते छ, राइट? एन्ड दे कम विथ द डिफ्रेन्ट इन्टरप्रिटेसन अबाउट द एलिफेन्ट. सो म तपाईँलाई के यहाँ एउटा चाहिँ मेटाफोर क्रिएट गर्न खोजिरहेको भन्दाखेरि, द मेटाफोर इज द्याट वी आर लाइक द ब्लाइन्ड ह्युमन्स, राइट? द ब्लाइन्ड पिपल. अब त्यो हाम्रो जुन एलिफेन्ट छ, त्यो एलिफेन्ट इज अ बिग बायोलोजिकल डेटा, राइट? त्यो बायोलोजिकल डेटा जुन जेनेरेट भइरहेको छ त्यसको के इन्फरेन्स गर्ने भन्ने कुरा चाहिँ इट डिपेन्ड्स अपन अस, राइट? सो बिग डेटा छ, देयर इज बिग डेटा, and how we want to interpret that data. अब what happens is that अब चाहिँ हामीसँग के faulty assumption हुन सक्छ भन्दाखेरि अब लाइक एउटा ब्लाइन्ड पर्सनले के गर्न सक्छ भन्दाखेरि त्यो एलिफेन्टको लेगलाई चाहिँ लिम्सहरूलाई चाहिँ फिल गरेर द्याट हि मे थिङ्क द्याट इट्स अ लग है त एउटा काठको चाहिँ एउटा चाहिँ लग हो भनेर चाहिँ हि क्यान इन्टरप्रेट द्याट तर के छ भन्दा द्याट इज अ रङ इन्टरप्रिटेसन अब त्यो चाहिँ अब होइन सो अब द्याट काइन्ड अफ मिस्टेक्स है त अर मिसइन्टरप्रिटेसन हेज टु बी अभोइडेड अब त प्रब्लम के छ भन्दाखेरि द्याट वी आर अ ब्लाइन्ड है त हामी ब्लाइन्ड छौँ हामीसँग त्यो सेन्सर इनपुट नै हामीसँग छैन सो देयर आर दिस बायोलोजिकल डेटा big amount of biological data and we have this challenge as well as opportunities so we can understand that this big elephant is a big एउटा चाहिँ organism हो so भनेर चाहिँ हामी यसलाई बुझ्न पनि सक्छौँ so we have that opportunity as well as that problem है त च्यालेन्ज त्यो पनि छ किन भन्दाखेरि देयर मेनी लेभल्समा इन्फर्मेसन छ जिनोममा न्युक्लियोटाइड सिक्वेन्समा राइट protein sequence map, protein interaction, pathway, systems, molecular imaging, molecular pathway, people, population, clinical, medical, lots and lots of information, and how to translate this massive amount of information so that we can generate a better understanding of human biology, right? So, के छ भन्दाखेरि so there was always है त यो बायोइन्फर्मेटिक्स कति हेल्पफुल हुन्छ कति हेल्पफुल हुँदैन भन्ने कुरा चाहिँ इट वाज अलवेज अ प्रब्लम बिकज वी जेनेरेटेड लट्स एन्ड लट्स अफ डेटा राइट सो एन्ड द फेज टू है द फेज टू वाज द इन्टरप्रिटेसन पार्ट एन्ड द प्रब्लम चाहिँ के थियो भन्दाखेरि so this researcher the researcher is saying that I think I found a corner piece so this big data out there right there's big data out there these 3 billion pieces right and now we want to make a meaningful information out of this data and I think bioinformatics has worked a lot right bioinformatics एकदमै contribute गरेको छ about the understanding of the biology so सुरुमा there were these misconceptions or there were these tools or techniques use गर्नको लागि there was some reluctance but what we have found that life can be understood in terms of bioinformatics principles we can better understand right so as we can see there lots and lots of data it's being generated and there is this astronomical accumulation of the genomic proteomics and the metabolomics data and we need to store this data analyze this data annotation पनि गर्नुपर्ने छ right organization systematization integration right अरू डेटाहरुसँग पनि यसलाई इन्टिग्रेट गर्नुपर्ने छ into the biological networks and the database systems and so यो सबै कुरा चाहिँ के हुनुपर्छ भन्दाखेरि there has to be this more or less a synergistic effort है त के हुनुपर्छ भन्दाखेरि so biology and computational science को चाहिँ there has to be synergistic effort so that we can build better networks better biological databases right and so that we can develop these tools better tools है त so that is the one of the major है त एउटा चाहिँ need चाहिँ यो छ so what is the summary summary is that bioinformatics targets to develop a methodology right there's a methodology analysis tools to explore large volumes of the biological data helping to store organize systematize annotate भनेको के what is the meaning of annotation के annotate गर्नु भनेको what is the meaning of annotation annotation भनेको चाहिँ के हुन्छ to add कुनै इन्फर्मेसनलाई चाहिँ कुनै कुनै छ कुनै एनोटेसन गर्नु भनेको चाहिँ फर्दर इन्फर्मेसन प्रोभाइड गर्नु राइट सपोज अब दिस इज द जिन एउटा जिन छ यो जिनलाई चाहिँ के गर्न सक्छु भन्दाखेरि म चाहिँ यसमा चाहिँ यो जिनको चाहिँ यो प्राइमरी डेटा छ भने योलाई चाहिँ म के गर्न सक्छु यहाँनिर प्रमोटर रिजन छ है त यहाँ प्रमोटर रिजन छ यहाँनिर चाहिँ एउटा ट्रान्सक्रिप्सन फ्याक्टर आएर बाइन्ड हुन्छ भनेर म चाहिँ आई क्यान एड एक्स्ट्रा लेयर अफ इन्फर्मेसन राइट सो द्याट विल मिन द एनोटेसन तपाईँको आफ्नो किताबमा पनि कुनै हाइलाइट गरेर इम्पोर्टेन्ट प्यासेजहरू इम्पोर्टेन्ट wordsहरू you can highlight that, and what are you doing actually you are annotating your information. Similarly हामी के गर्न सक्छौँ भन्दाखेरि जेनेटिक इन्फर्मेसन्सहरू र इन्फर्मेसन्सहरूलाई चाहिँ we can annotate that, right? अब यो एनोटेसन भनेको कुरा कस्तो हुन्छ भन्दाखेरि lots of people can contribute at that information है त, right? डेटाबेस बनाउन सकिन्छ, right? And then different scientists working independently they can have that information है त, and collaborate with one another. And that's the very important part in bioinformatics, to learn to collaborate, to learn to develop systems, right? Tools that can help the scientists to collaborate one another. So to visualize, to query, to mine, and to understand and interpret the complex data volumes. So it uses conventional modern computer science, cloud computing, statistics, and mathematics, as well as pattern recognition, reconstruction, machine learning, simulation, and iterative approaches, and molecular modeling and folding algorithms. So lots and lots of disciplines, right? Computational disciplines are used in bioinformatics. So bioinformatics develops and utilizes computational algorithms to understand and to interpret biological processes based on genome-derived molecular sequences and their interactions. So bioinformatics focuses on providing practical tools to organize and analyze basic genomic, proteomic, and other omic data, including sequence analysis and its visualization. So bioinformatics tools widely used for studies related to genetics, genomics, biochemistry, physiology, biophysics, agriculture, medical and environmental sciences, as well as evolution, system biology, and artificial intelligence. So एकदमै धेरै ठाउँमा चाहिँ यसको के छ भन्दाखेरि टुल्सहरू चाहिँ वी क्यान युज इट है, त्यसको रोल्सहरू स्कोप्सहरू एकदमै धेरै धेरै छ, राइट? आज हामी यति पढ्यौँ है त, वी स्टडी टिल हियर. So do you have any queries?